Okay, we are now moving into the last part of this session, and we have 3 presentations. And first out is Buchholz et al., but the presentation is given by Jonna Tomkiewicz. And the title of this presentation is Estimation of Baltic herring oocyte dynamics and fecundity using stereological methods.
I am here to give a presentation for Rikke Buchholz, a PhD student of mine who unfortunately had to cancel last minute. The focus of the study is estimation of Baltic herring oocyte dynamics and fecundity using stereological methods. Involved in the study is also Johnny Andersen, Jens Nyengård and Hans-Jørgen Gundersen from University of Aarhus in Denmark. The objectives of the study is to describe the oocyte dynamics and fecundity in Baltic herring using stereological quantification of oocytes. We want to estimate the size frequency distribution of 4 oocyte development stages: primary growth oocytes, cortical alveoli oocytes, early vitellogenic and late vitellogenic oocytes in relation to maturation stage. We want to estimate potential fecundity in relation to length and maturity stage. We want to relate fecundity to the mean developing oocyte size, and we want to see whether we can distinguish first and repeat spawners. We look into factors potentially influencing fecundity, Fulton's condition factor, and state of maturation and ovary mass. First, the reproductive strategy of herring. The strategy is relatively simple. It is an Echinoparous species. It has group synchronous development, and it is a total spawner. And we look at, where do I point? There. Yes. We look at 4 stages. We look at juvenile, we look at and 3 maturation stages. That is stage 1 to 4. In the first maturation stage, the juvenile, the ovaries are dominated by primary growth oocytes. In the stage 2, they are dominated by cortical alveoli oocytes. In stage 3, it is the early vitellogenic stages, and in stage 4 is the late vitellogenic stages. The sampling was conducted in the Baltic Sea, more in specific in the Bornholm basin of the Baltic Sea. That is this area here, and it was conducted on board of our research vessel Dana in March 2008. Throughout the study, we used design-based stereology, in particularly the fractionator method, which is used to quantify the number of oocytes in a known fraction of the ovary. The systematic uniform random sampling source is the basis of this sampling. On board of the cruise, herring were sampled at random from the trawl holes and fish data were recorded. The ovaries were weighed and preserved, and in the lab, 1 ovary lobe was randomly selected and embedded in paraffin. The lobes in paraffin were cut into 8 blocks using a random start. The blocks were sectioned to obtain dissector pairs using different distances between the sections for the different oocyte types, and depending on their size, this is creating different volumes, but in each volume, the oocytes can be counted using this method. In the histological analysis, we are using an 8-slide stage and this image analysis software. That is a software from Visiopharm, and Visiopharm is here also represented by Johnny Andersen, a co-author of the study, and he will have a booth in the poster area that you can visit and ask further. The histological analysis, as I said, used physical dissector. Oocytes counts are made using the dissector pairs. We use the nuclei as the counting objects, and in this method, we have a whole section that is the entire ovary sectioned, and random sections are then used for the imaging, and we are comparing 2 sections with a known distance. In this method, we are then counting which oocytes are disappearing, or actually the nuclei, which nuclei are present here, but not here. We call this the lookup section, and this a reference section, and we are using a counting frame. We do that both backwards and forwards, so we use both this one and this one to see whether an oocyte has appeared or disappeared. Then we divide the total number of oocytes with 2, and then we get an estimate of the number of oocytes of this size in this particular volume. We also correct for shrinkage. We have looked at fresh tissue, and we are looking at the preserved tissue, and we have made a correlation, which very nice linear relation, which do that we are able to calculate from the fixed or from the slides to fresh oocyte diameter. That is called the nucleator method, also in the WIS software, is used to estimate the size of the oocytes. And this is used in the size frequency distributions, and here you're having the results. The first example is how does actually the size frequency distribution look in a juvenile herring, the stage 1. In this case, we have only primary growth oocytes, which have a distribution where the largest oocytes are just above 200 micrometers. If we then enter the early maturation and the recruitment of the oocytes which are to be spawned this year, we can see that in the early part of the stage 2, we are having recruiting oocytes. We see how the CA oocytes are budding off from the primary growth oocytes. And this distribution overlap here disappears as the ovary develops. We are obtaining a hiatus between the primary growth and the cortical alveoli oocytes as the oocytes they develop. The further development is creating a larger hiatus between the primary growth and the vitellogenic oocytes, and in the late vitellogenic stage, the final maturation, we see here as typical of a group synchronous species, large yolked oocytes and primary growth oocytes. The vitellogenic oocytes are here between 750 and 950 micrometers. If we now change this pattern of the size difference, the size frequency distribution into cumulated oocyte size distributions, we see first here the primary growth oocytes. They are combined from the 4 different stages, and we see here the primary growth oocytes in the stage 1, the juvenile, are slightly smaller than in the other stages, but there is no significant difference between the size in the different stages. If we then look at the primary growth oocytes and the cortical alveoli oocytes, there is an overlap in the range between 190 micrometers and 270. This means that for analysis of fecundity, we do not use ovaries where the cortical alveoli oocytes are having a diameter below 270 micrometers, because in those cases over here, they will not be fully recruited. If we look at the potential fecundity in relation to length, we observe that the fecundity is increasing with length, just as we would expect, but we also see that we have a higher fecundity in the early stages than we are having in the more mature stages. This indicates that there is a downregulation of fecundity from the early stages until the late stages. This one is actually not correct. It is actually, these 3 stages are stages which are having both cortical alveoli and early vitellogenic oocytes, and we can see that they are intermediate. Another peculiarity we noticed was that when we look at the cortical alveoli, we had some kind of deviation from the prediction in the curve. We looked a little bit further into that, and we discovered that we had a mixture of first-time spawners and repeat spawners. In the cortical alveoli stage, they are easy to recognize because the first-time spawners are having a very thin ovarian wall, whereas the repeat spawners are having a very thick wall. Later in the stages, this is more difficult to see, but in general, those ones below 20 appeared to be first-time spawners, while those ones, about 20, appears to be repeat spawners. So if we now distinguish, we actually saw that the larger oocytes were related to first-time spawners, whereas the smaller size of the cortical alveoli was related to repeat spawners. If we now look at the potential fecundity in relation to oocyte size, we again have that we have here the repeat spawners and the first-time spawners. And what we see is that the potential fecundity of the repeat spawners is much higher than is the case for the first-time spawners. If we also include the maturing stages with the vitellogenic oocytes and we look at first-time spawners, they also tend here to be less fecund than the repeat spawners, which is partially related to the size of the fish. We looked at condition in relation to development to see whether there was a relationship between the Fulton condition factor and the fecundity and the development, but we found out that when we looked at the increase in oocyte diameter and also in the gonadosomatic index, that there was an increase in the Fulton condition factor with the development of the ovary. So actually, the fish tended to gain weight with increasing development, so they were probably eating themselves to the extra weight, also known from other species. If we look at these repeat spawners with the cortical alveoli, they are here in the circle. And these repeat spawners, they actually had both a very low condition and very small oocytes. And we wonder whether these fish may be skip of spawning or they could belong to another population. We are now looking at the oocytes, at the otoliths, to evaluate whether it is possible to see previous spawning check marks and from the shape identify the stock. To conclude from the study, the oocyte recruitment, we were able to distinguish between the primary growth and the cortical alveoli oocytes, which overlap in the size range 190 to 270. And from those distributions, we could see that the minimum cortical alveoli diameter for fecundity analysis should be 270 micrometers, and that gave us an exclusion criteria for specimens in the study. In relation to fecundity, we had a clear relation to the maturation state as well as to the fish length. We saw a down regulation which continued throughout the ovarian development. And what we experienced was that the ovary mass, where did that actually go, that picture? Predicted potential fecundity within the maturity stages. The Fulton condition factor was positively correlated with GSI and oocyte development, and repeat spawners had a relatively low condition factor in the recruitment phase, which might indicate that they were skip of spawning stages. The stereological method we used, we found well suited to quantify oocytes of all development stages, and they had a low degree of noise. You could not see that, but we wanted to include the coefficient of error, but the coefficient is so low that you would not be able to see it in the symbols. With this, I will thank you for the attention, and this is Rikke, who could unfortunately not join us.